I'm going to get us started. Thanks for joining us. I know it's sunny and gorgeous outside. Today we are joined by a virtual presenter, but has a lot to say. I wanted to get him here. This is Brian DiMENTO online in the box. He's going to talk today about road runoff and tire toxins. He's with the University of Alaska in Anchorage, and he is actually currently in Anchorage. So Brian, go ahead and take it away.
Thanks for having me. Thanks for coming, everyone. I wish I could be there in person. I have not been to Cordova yet, but someday. Still branching out from Anchorage. I'm originally from Massachusetts, actually, and grew up on the coast out there, doing lots of boating and fishing and hunting and everything outdoors possible. And got excited about the opportunity to move to Alaska 2.5 years ago, and have been loving it and doing as many adventures as possible every weekend. So excited to share some of my work, but I figured I would start by kind of telling you a little bit more about my path to get here. There we go. I got my PhD in oceanography, actually, even though I haven't really done a lot of oceanography since then. But my PhD mostly focused on mercury in the ocean. So another toxin. Growing up fishing, my father was in the seafood industry. It was kind of something I was aware of for a long time, and I kind of fell in love with chemistry in college and was excited to do some sort of applied chemistry to the environment. And mercury was kind of a topic that stuck, and I wound up pursuing for my PhD and got to go on a couple of research cruises. This is a picture I took from the equatorial Pacific, quite different than the Arctic, but doing some work there, and also work around New England, like the East Coast of the United States, studying all aspects of what is a rather complicated mercury cycle. But you'll see, like 6 ppdq, mercury is found in really low concentrations in most of the environment and in the ocean. But in the case of mercury, why we cared about it was because of how much it accumulates, bioaccumulates, or biomagnifies, we say. The increase in concentration as you go from the water to humans, or to fish, to seafood, is something like 10 million times increase in concentration. So tiny amounts in the environment can still have an impact. And we were studying this complicated mercury cycle. I kind of used these complicated schematics a few times here just to show how complex and how many different processes we might look at. For one process be like air-sea exchange, that's mercury going from the water to the air, or it coming back from the air into the land or water, that's atmospheric deposition. How it gets into fish, how the more toxic form of mercury, which is methyl mercury, is formed and degraded. That's kind of one of the recurring themes of my research that I will mention in multiple forms, is photochemical reactions, so sunlight mediated reactions. That was one chapter of my dissertation, looking at how sunlight breaks down methyl mercury. It was also my introduction to the Arctic. I didn't get to go on this research cruise, but we sent instrumentation on the Coast Guard cutter Healy from Alaska to the North Pole and back. And we were looking at how sea ice impacts the accumulation of mercury under the ice. You actually get a lot of buildup of that gaseous form of mercury under the ice, which has implications for what winds up in seafood. After that, I kind of switched to more organic, or in this case, carbon-based organic, not organic as in pesticide-free. A pollutant called a short-chain chlorinated paraffin, which is one of those things that no one's heard of, but it's everywhere. And it's just this long carbon chain with chlorine molecules on it, or atoms on it, kind of similar to like a PFAS you've heard of probably, that it's really good at its job. It's like an industrial lubricant, and it's a plastic additive, and it's used all sorts of places. Turns out it was toxic. Turns out it got everywhere. And we wanted to understand how it broke down in the environment. So we were doing some photochemistry studies using this picture in the background here is a common instrument we use that's called a solar simulator. It is basically a really fancy light bulb that mimics the sun, and we can turn it on as bright as we want for as long as we want, and kind of control our reaction and how Depending on how fast it happens, it's a lot easier to do the work than to put stuff outside to study sunlight. And that's something that I've continued, and we'll talk a little bit about today with 6-ppdq in Alaska. So another topic I'll talk about in terms of these photochemical reactions is these reactions with other reactive species in the environment. Free radicals may be a familiar term, and I won't get into all the details here, but one thing we like to do is understand how the reactions occur so that we can kind of predict how they will, like the extent they'll happen in different environments. That's something we did with 6-ppdq as well. After that, I went on actually back to the college where I did my undergrad degree. And again, one of the projects I was studying, I love this picture. This is called a chemiluminescent reaction. It is a chemical reaction that produces light. And we can use that light, how much light is produced, to quantify how much of the compound is present. And that's actually the reaction happening outside of the instrument we use, but it makes this really pretty blue light. Not important otherwise, other than being interesting in this case, but this is the place where I started getting more into kind of a water quality, local environmental issue interest. And one of the big projects I worked on there was a lake water quality study. The chain of lakes there were suffering from excess nutrients and had these nasty algae blooms. That background picture is a little algae monitor that floats on the surface. It's like a little frisbee, but you can see it floating in the green scum that once was a beautiful lake. I spent a lot of time outside studying these lakes and why these blooms happen and what we could do about them all times of the year. So through the ice or in the summer. There's always an excuse for me to go ice fishing in the winter too, because I already had the holes drilled and everything. So then 2.5 years ago, I got the opportunity to go to Alaska, joined the chemistry department at UAA, and the very first project I worked on was studying 6-ppdq, and it's kind of giving away the punchline for what we're going to talk about, but worked on a bunch of other projects since then, or during and since then, looking at other toxins like microplastics and saxitoxins, other Alaskan relevant pollutants. But the 6-ppdq question has been something I've been interested in since starting here, and it's been a challenge to get funding to work on it. My position is entirely funded by grants, so it's hard for me to work on things we don't already have funding for. But it's hard to get funding for things when you don't have data to prove why it's important to study them. And 6-ppdq is a good example of that in our lab. So let's get back on track here. So what is 6-ppdq? Why do we care? How was it discovered? Not sure how many of you are familiar with the topic or have heard of it before. If I was there, I would ask for a show of hands maybe, but I can't see everyone through the camera. So just tell the story. Started or was discovered in the Pacific Northwest a few years ago only. The first paper came out in 2021, but after the Seattle area spent billions and billions of dollars on restoring salmon habitat and getting their coho back to the local streams, which was successful, they started seeing the salmon die off before they had a chance to spawn. And they didn't know why. They called that urban runoff mortality syndrome because it always happened after it rained. And what a group of scientists decided or studied was what was in that rain washing off the roads into the streams that was harming the salmon. There's a lot of other pollutants that are in road runoff, petroleum products, brake dust, things from tires wearing on the roads. So there's a lot of potential culprits for what could have been killing the salmon, and it could have been a lot of things combining together to kill them. But whatever it was doing a really good job of killing the coho in just hours after a rainfall. Especially in the urban centers. So I really enjoyed hearing one of the lead scientists talk about this, getting into the chemistry of how how they figured out the compound. It was a lot of work, but ultimately they identified this tire additive 6-PPD, which is the chemical. I don't know if you can see my mouse here. Chemical on the left, which is a ubiquitously used tire additive, and it has been used since the 60s. It's great at what it does, which is sacrificially reacting with ozone, which is formed as a byproduct of combustion in the atmosphere. Different pollutants from combustion can react also photochemically, actually, to make ozone. And ozone reacts with 6-PPD, the tire additive, and it just so happens to form this incredibly toxic compound, which is abbreviated 6-PPDQ. The Q stands for quinone, and that's this kind of group in the center with the two oxygen atoms opposite each other. That compound, 6-PPDQ, was not known before this time. They actually used a fancy high-resolution mass spectrometer to identify its structure, didn't know where it came from until they started looking at other compounds that existed that might be in road runoff and discovered that 6-PPD was something tire industry used all the time that people didn't really know about, and they theorized that was the source of the 6-PPDQ. And it's really good at its job. It does something that's called blooming, where as the compound reacts at the surface of the tires, new compound migrates to the surface. So it can sacrificially react with the ozone and protect the tire polymer. That actually results in, you might have seen like a reddish brown color on the surface of your tires, and could be brake dust, but sometimes it actually is the 6-PPDQ or other tire additives that oxidize on the surface of tires and keep your tire from dry rotting. You can see that picture showing what tire rubber looks like without the additive. It just rots too quickly and it's unsafe to drive on for very long. So because of that, the tire companies can't just stop using it. It's more complicated than that. Obviously, the EPA would love to ban chemical that's killing salmon, but we need to find an alternative to the compound first, and the U.S. Tire Manufacturers Association is working on it. It's a slow process, but they've identified a bunch of alternatives. But not only does the alternative need to work really well, it also can't be toxic. And not only can the additive not be toxic, but anything it forms can't be toxic either. And that was maybe an oversight in this case, or a common problem where you say, oh yes, the additive goes away, but what is it forming when that compound goes away? And is that toxic? And you can continue that down the line as things degrade.
Can I ask you a question?
Yeah.
So, because we have a lot of gravel roads here and they have a lot of paved roads there, has there been any study to say it happens more when it comes in contact with asphalt or cement roads or gravel roads?
Yeah, generally a more impervious surface contributes more to the compound winding up in the water. I don't know if I've seen the data on how much is being formed in the first place, but the compound does tend to stick to like soil and other organic matter like that before it kind of gets into the water. So the places where it's the worst are where an impervious surface runs off directly into a stream, which happens in cities a lot. So other areas, if there's some sort of buffer of soil or vegetation on the side of a gravel road, then it's not as bad. I'm sure there's a lot more research that needs to be done and data that could be collected on that kind of situation. But the worst cases are always more impervious surfaces where the water doesn't have a chance to kind of percolate through the ground at all.
Thank you.
So this compound, 6-PPD quinone, turned out to be one of the most toxic chemicals known to aquatic life, on par with all the other pesticides and things that have ever been used. We quantify toxicity with what's called an LC50. It's the lethal concentration for 50% of test organisms over a 24-hour exposure. And for coho salmon, determined at UAA, that level was 67 parts per trillion. which you'll see abbreviated as either PPT or sometimes nanograms per liter. A nanogram is 10 to the power of -9, 1 billionth of a gram per liter. And that's a part per trillion is roughly equivalent to a single drop in 20 Olympic-sized swimming pools. It is a tiny concentration. That makes it incredibly deadly, and it actually is really only deadly, or only that deadly, for cohos, for silvers. It doesn't really seem to affect the other salmon species. There's a few other species in the trout families that are susceptible, but none nearly as susceptible as coho. And we don't know why still to this day. Lots of people have been working on it, and no one has quite found an explanation for why coho are more susceptible than other species. I'm going to get back into some of this toxicity work later in the talk, so I have a chance to see some more of this work. Because those concentrations are so low, they're really hard to measure. And traditionally, this approach or measuring these concentrations required collecting a rather large volume of water, like 250 to 500 milliliters. That's like a couple of cups of water, and concentrating it down into a small volume. And that meant this method was more sensitive. That meant it was a lot harder to collect samples, more time, and more time means more money, which the estimate I saw was that it costs a lab about $500 to analyze a single sample. So we really didn't have a lot of data in Alaska over the past few years once this problem was discovered. There was a handful of measurements, like 6 or 8 or 10 maybe a few years ago, that showed that this compound was present in Anchorage. And it was definitely detectable in storm drains, but the stream measurements were all pretty low still. So since then, not a lot of measurements had been made. But that changed last summer when I had the help of this National Science Foundation Research Experience for Undergraduate intern in the picture here, Elizabeth, helped me out last summer with drastically increasing the amount of data we have of 6-ppd quinone in Anchorage. So this is her doing that extraction, the traditional extraction procedure. It's called solid phase extraction. The water gets pumped, or actually through these cartridges, which have absorbing material that will trap the 6-ppdq, and then we can remove it from those cartridges using a different solvent and get a much more concentrated sample. Worked great for really sensitive measurements, but every sample took hours to process, and we can only get so much data. And when I didn't have dedicated funding to work on this, it's not something I was able to devote my time to. So thankfully, we were able to get more data last summer, and also kind of work on a newly published method that skips this step. Requires a more sensitive instrument, but thankfully we had just purchased a new $1 million LCMS, liquid chromatography mass spectrometry, in the asset lab at UAA, which facilitated doing these direct measurements at concentrations that were low enough to be meaningful. The method was sensitive enough to be useful for us now, and it went from several hours for analyzing one sample to like 10 minutes to prepping a sample. So we were able to get a lot more data finally. We took the approach of looking at storm drains and stream sites downstream from those storm drains at a bunch of sites around Anchorage, mostly Ship Creek, Chester Creek, and Camel Creek, if you're familiar with Anchorage. And we used the municipality's storm drain maps to kind of predict hotspots. They have mapped out the drainage basins of these storm drains, and we could predict what ones included large amounts of impervious surfaces, paved roads, and major roads with lots of traffic, obviously, would result in more tire wear particles forming, and those would leach out 6-ppdq when they got into the water. So we collected samples all around Anchorage. If you remember, last summer. I don't know what it was like in Cordova, maybe different in Prince William Sound probably, since it tends to be wetter. But in Anchorage, it was a very dry June and July, or May and June and half of July probably. This is a compound that is released on roads, but it needs rain events to get into the stream. So we're going to see that these rain events, these storms, are really important to transporting 6 ppdq into the water. And these two figures represent all of the samples we had analyzed over the first 2/3 of the summer. So every one of these bars is a single water sample, and all of the blank spaces are samples that we couldn't detect 6 ppdq in. The concentration in parts per trillion is on the vertical axis, and then that LC50, the lethal concentration for 50% of individuals, is in that red dashed line at 67 parts per trillion. So notice that the scale on the right side, where the storm drain water is plotted, is much bigger. There's a lot more 6 ppdq directly in the storm drain water, but the salmon aren't swimming in the storm drains, they're swimming in the streams. And that plot doesn't take into account how much flow is in those storm drains. Some of those are just a trickle, some of them are flowing stronger. It depends on whether or not it rained recently. But what should stand out to you is that there are few data points on the right side of those plots that are quite high, and that's kind of what I'm going to focus on next. So after that dry summer, suddenly on July 25th, which was like the very last week that Elizabeth was staying in Alaska, we had in the forecast a really strong storm coming. And I made sure to get myself to one of our sites on Chester Creek to be ready to collect samples, because we know that for other pollutants in 6 ppdq, we have what's called a first flush phenomenon, where just as it starts to rain, the concentration of the pollutant is higher in the water, because those dirty streets that are all dusty from all the previous month of no rain, all of that gets washed out. And the more and more it rains, the more the streets get washed off, the cleaner they get. So their concentration goes back down. So we really wanted to capture that first peak in concentration. These two pictures show our storm drain site. And it's the same culvert in the left and right image. The left image is from 9:35 in the morning, and then a half hour later, a little more than a half hour later, that's the same culvert or storm drain flowing on top of the culvert, which is the stream itself flowing under the Seward Highway. The flow went from kind of a trickle to a deluge, and the water went from nice and low and clear to high and brown. And I was collecting samples the whole time. You can't tell, but it's absolutely pouring out. I was getting soaked. And here's what the data look like. So I will first point out this dashed line, and that is actually the USGS stream gauge. So it's the water height, and you can see the water level before the storm, and it started raining hard around 9:30 in the morning, and the water level rose rapidly. And you can see how the concentrations plotted followed. So the top two lines are the two storm drains that are nearby. And one of them you can see has a concentration of 3,000 parts per trillion, really high amounts of 6 ppdq. The one of interest, obviously, is the stream itself. So these samples were taken kind of downstream from the storm drain, so the water had a chance to mix. Earlier in the week, that same stream was something like 5 parts per trillion. And the first sample I took is right as it started to rise. And that one was around 20 parts per trillion, which was already the highest concentration we had measured in the stream in Anchorage. But that very next sample, as the water rose, the concentration shot up over 200 parts per trillion, which is way higher than the LC50. And then it stayed above the LC50 for the rest of the day. So if there was a salmon present in the stream at that time, it wasn't going to be very happy. The amount of time it takes for the salmon to die well, kind of depends on the concentration. The the standard for toxicity is a 24-hour exposure, but in the Pacific Northwest, they saw them dying within hours of a big exposure. So it's possible. I did not observe any salmon and did not hear of any mortality, but it's possible that there were salmon present here or downstream that were impacted. Unfortunately, this is only one location for one storm event, so it tells us that this could be a problem, but we don't know how much of a problem until we get a lot more data. Upstream from here, I expect the concentration to not be as high because the water hasn't had a chance to accumulate from all the storm drains in the city. That was kind of the next step for me, was kind of covering a larger spatial area. And after July, it gets a lot rainier in Anchorage, so that storm was probably kind of like a perfect storm of a long dry period where a lot of tire wear particles accumulate on the road, and then suddenly strong rain washes them into the streams. In late summer, Anchorage gets lots of rain, so doesn't have a chance to accumulate as much. But there was one other storm event that I was able to go out and sample in early October, which wasn't as kind to me in its timing. I mentioned I like to capture that first flush, the beginning of the water rising. So these are two stream gauge measurements. Again, it's the water height. You can see as it starts to rain, the water goes way up. But if you look at the time on the horizontal axis, that was at midnight when that rain started. So I was not out sampling in this case. I sampled before the rain and after the rain, which previous data tells us that probably missed the peak of the concentration, but it was still interesting to try to collect samples from kind of a downstream and member upstream into the city for both Chester and Campbell Creeks. So that's what the next data are going to show. Top figure is Campbell Creek, and the concentration all the way down at the lake was in the 27 part per trillion range, and it went down as you went upstream. So that's kind of what I expected. There's more and more city street inputs as you go downstream, so the concentration rises. Chester Creek was generally showing that same trend, although there was one sample I collected that was on a separate tributary of Chester Creek that showed higher concentrations. That high concentration was only in the 30s. It was not the 200 part per trillion we saw in the previous storm, but if 67 parts per trillion can kill them, I can't imagine that 30 is good for them. So again, we didn't observe mortality here. This was late in the season at this point, but it's important to keep in mind that while the original observations were adults, there are always juveniles in these streams. So they are susceptible as well. And we need to be aware of what concentrations are present at all times of the year. So this data I shared with the 6 ppd quinone work group that we have. That's people, anyone in Alaska involved in 6 ppdq research, along with some tire industry people, get together once or twice a year and discuss the latest findings. And I shared this data, and it got the interest of the DEC, and they wound up giving us some money to continue this work for the next year, fiscal year actually, until June 30th. So I finally have some more dedicated funding to do some of this work. That funding became available kind of in the winter, and it wasn't a great time to sample. So we're going to be ramping up efforts coming into this melt season and into the summer again to see what we find. Here's the data just showing how that concentration went up as the rain or as the storm passed. So it's a big jump in concentration, even though we didn't surpass that LC50. Some other work we've done that we're hoping to do more of is looking at snow dumps. So in the winter, all of that snow from the roads and all that road debris gets piled into specific spots around Anchorage. And clearly that's going to have some tire wear particles in there amongst all that gravel that gets scraped up from the roads. So we've done a little bit of sampling of the meltwater and haven't found much yet, but actually was just emailing with someone today about doing some more this year and trying to capture that a little better. So stand by for more updates on that front. But like I said, coming up into this spring and summer, with that funding we got, we actually now have a couple, we call them auto samplers. They're these funny garbage can looking devices that are just pumps that can, with a fancy robotic arm, that can fill water bottles in a sequence, with a program to kind of at even increments collect water during the course of a rain event. So we can deploy these around the city when we're expecting rain and have them just automatically collect water every half hour or so. And we can really capture the changing concentration over time. So that's one thing we're doing to help increase the spatial coverage of our data, along with the temporal, the time coverage of sampling, so we don't miss those peaks if they occur overnight. So we're hoping to get a lot more data, and that's ultimately hopefully going to support some more work to actually fix this problem. And I'll kind of come back to this at the end, but obviously the next step is a real solution for the problem. We know it could exist in Anchorage. I'm still trying to figure out how bad it is and where else it could be a problem. So I've been in talk with other people around the state. We're going to do some more work on the Kenai. Hopefully doing a little Susitna as well that has a lot of road system access. And we have a little bit of money at the moment to provide sampling equipment for people to collect samples and send them to us. And I can cover to a limited extent the analysis costs. So trying to extend our reach on where these samples are taken. I wanted to spend a little bit of time talking about some of the other work at UAA as well while we're on the 6PPQ topic. But I can pause here if anyone wants to ask questions about some of the data I just showed.
Yeah, I got a couple questions.
Yeah.
One is, has anyone looked at the fish to see what the mechanism of death is? You know, like, is this compound stopping the oxygen from being transferred from the gills or whatever? And then the other question is, how persistent is it in the environment? Will it eventually form into something else, or does it just settle to the bottom, or what?
You're setting up really well for the rest of my talk.
Okay, there you go.
Yeah, the mechanism of toxicity is, I am not a toxicologist or a physiologist, and I am still shocked of how little we seem to know about how it's impacting the salmon. It seems kind of like other species just have a better way of breaking it down that the coho lack for some reason, but I haven't seen a really conclusive study of what exactly or how exactly it's killing them. So that is still kind of an unknown, surprisingly. Persistence in the environment so far seems promising. It is not especially a persistent compound. I mean, this is definitely a lot of a solution of the solution to pollution kind of situation where it's just getting flushed out of streams. It's not sticking around, but it is also a compound that likes to stick to soil and organic matter and vegetation. So water that flows through vegetation and wetlands and things like that seems to do a really good job of removing it, whether it's just sticking and eventually being biologically degraded, or one of the processes I'm going to talk about that I've mentioned multiple times leading up to this is photochemical degradation. And that's a process, another common way that a pollutant breaks down the environment. And 6-PPDQ is also really susceptible to that. In a really brown, nasty, high-flowing, sheltered stream under trees, that's not really necessarily the most likely way for it to degrade, but it still is promising that it's not going to be a really persistent pollutant like PFAS or something like that, or mercury that doesn't really go away. So it doesn't seem like a problem that will get worse and worse. It's a problem that is short, it comes on hard and strong every rainfall and then dissipates. Any other questions?
I was wondering, when you mentioned earlier that, well, you talking about how it affects silvers mostly.
Mhm.
So was that also tested in the lab or just, in the wild where we're seeing visually? Because the reason I guess I ask is because silvers are so good at coming up these little streams into, all kinds of places where kings and reds would be going up the bigger waterways with more water.
Yep, that, I mean, again, also leading into what I was going to talk about next, but it has been tested in the lab. UAA specifically with reds and kings. I don't know that people have done chums or pinks. Maybe there's less concern with them in the city rivers and the urban rivers. Maybe someone else has, but not in Alaska to my knowledge. But at UAA, we studied silvers, reds, and kings as well. So, and they are shockingly not susceptible.
Interesting.
And that's what I was going to talk about next. So the toxicity aspect is something that's been done a lot at UAA for the past couple of years, led by Jonathan Stesick in the biology department with some other scientists and lots of undergraduate help. They were looking at the acute toxicity of 6 ppdq, so what level is deadly to salmon fry, but also what happens when they're exposed to sublethal concentrations. So like I said, if 67 kills them, 30 can't be good for them, right? So they kind of looked at some aspects of that question. And I was also going to talk about our photochemical degradation project that I was involved in, and how 6 ppdq, or one of the ways by which 6 ppdq degrades. So here's your lab studies right here. To study the toxicity, they got salmon fry from the hatchery in Anchorage and Golconda, I think. And they basically put them in a series of tanks at different concentrations and observed how many died over a 24-hour period. For coho, because that was already studied, they had a target range to expose them to. That was 15 to 150 parts per trillion. Sockeye and Chinook, which hadn't been shown to be susceptible, they went way, way, way higher, 125,000 parts per trillion, and saw no mortality in that case. So that was the figures I kind of hinted at earlier or showed earlier. These LC50 plots where you have concentration along the horizontal axis and mortality along the vertical axis, and you can plot how many of the individual salmon fry died over the course of the 24 hours versus the concentration, and then extrapolate to where 50% of them died, and that's how they got that 67 number. On the right side, you can see the sockeye and Chinook plot, and none of them died all the way up to that incredibly high concentration. So, like had been seen elsewhere, the Alaskan salmon's strains, I don't know what you want to call them, same species, but salmon that actually came from Alaska kind of showed similar toxicity to salmon elsewhere. One of the other tests that was done was looking at lower exposures on temperature tolerance. So salmon are cold water species. They don't like the water to get too hot. And this study basically put a bunch of salmon in a tank and slowly warmed up or exposed them to 40 part per trillion 6 ppdq, and then slowly warmed up the temperature and looked for the salmon to flip over, lose their equilibrium, or their ability to sort of keep upright. And a 24-hour exposure to 40 part per trillion resulted in a lower temperature, a CT max, critical thermal equilibrium. So they became more sensitive to warm temperatures after 6 ppdq exposure, which is not great if our streams in Alaska are warming up. Another one is the impact of 6-ppdq on their ability to take up oxygen. And that study was basically similarly exposing them to 40 part per trillion 6-ppdq for 24 hours, and then basically chasing them around a bucket for a while to get them all tired, and then measuring how much oxygen they are taking up. And exposing them to 6-ppdq reduced how much oxygen they could take in after exhaustion. The resting breathing rate, respiration rate was unaffected, but they were basically less fit after being exposed to 6-ppdq. So a couple other reasons or a couple other mechanisms other than dying that showed that 6-ppdq at lower concentrations could be harming the wild salmon. And that was all species specific. They did that work with the sockeye and chinook as well. And all these effects varied, and coho were more sensitive than others. So definitely a species specific sensitivity. That's not my field, but I'm happy to attempt to take any questions on that topic.
You should be smiling because this should be.
The last topic is our photochemistry, and that is my field. I stole these slides from a colleague, so they are a little complex, but I just wanted to point out some of the different aspects of what we study and why when we're looking at environmental fate of 6-ppdq and why we study photochemistry. So we're talking about what happens to the 6-ppdq when it goes, when it's released into the environment. Well, it's on the roads, it deposits onto the roadways and the surrounding areas. And also when it rains, it runs off into the water. And when it's in the water, it can potentially volatilize from water to air. So there's all these different phases we can study, all these different reactions that can happen in each of those phases. And the one we like to study is this photolysis, this photochemical reaction in the water. Obviously, the water is the important one to study here because it's where the fish are. So we are looking at kind of two processes. We call them indirect and direct photochemical reactions. A direct photochemical reaction occurs when the compound itself is capable of absorbing light that reaches the surface of the Earth. So this figure on the right shows the solar spectrum. These are wavelengths of light across the horizontal axis, and then the intensity of the sunlight in these jagged lines. And then this one big peak represents how or what wavelengths of light 6-ppdq absorbs. And the fact that there is overlap between those two spectrum tells us that 6-ppdq could directly react after absorbing light. The energy from the light could cause it to break down. We can also have other pathways where other compounds that are dissolved in water, we call them dissolved organic matter for one, or colored dissolved organic matter, CDOM. That's like the stuff that stains water tea colored. You have a lake that looks brown or a stream that looks brown. All that stuff in the water is also potentially photochemically reactive and can produce kind of these reactive intermediates that can go on and react with 6-ppdq. So we like to study both of those processes, but we do it in the lab. So we use that solar simulator, that artificial sunlight, to study all these different arrows and see how fast they happen and which ones are important. And that kind of all builds up to telling us how long 6-ppdq could last in the environment. And as you change the water chemistry, you change the pathway, and you can predict in different waters and in different latitudes. So as the temperature changes and the sunlight intensity changes, how is this reaction going to change? And that was the first project I worked on, and that's now published research. I won't get into all these arrows here, but basically 6-ppdq does degrade relatively fast by photochemistry if you have the right conditions. If you have nice clear water and sunlight, which we have in the summer at least, you can break down this compound. Another question that was brought up was, or I brought up, and I think it was also brought up, of what about the other things that are formed when 6-ppdq degrades? We saw that 6-ppdq itself is a transformation product. It was not the additive, it was what formed when the additive reacted. And that thing that formed happened to be really toxic. So we have instrumentation at UAA. It's a high resolution mass spectrometer that is sensitive enough to identify or predict chemical structures based on their specific exact mass. And that is what this really complicated figure of all these different chemical structures represents. It's using a similar process like we used to use for the environmental 6-ppdq analyses, solid phase extraction, to concentrate the products that form after we do those photochemistry reactions. And we can try to predict what all those chemicals are. And then if there are some that seem really common, those could potentially be taken and studied further to see how toxic those are. Are those toxic as well, or are they turning into more harmless things? And what we saw made us pretty confident that for the most part, it seems like as 6-ppdq degrades, it's going to become less toxic just based on the different types of structures we're seeing that form. But it's another area for future research. You can see it's a very, very complicated pathway. So takeaways from this work, this is a now published paper I could share if you were interested in learning more about photochemistry. But we think that photochemical reactions are a major way with which 6-ppdq could be removed from the environment under the right conditions. It happens faster when the temperature warms up. But those kind of indirect reactions I talked about are also seeming to be really important when the temperatures are colder, which is common in Alaska. So we feel pretty good that 6-ppdq is not going to linger around, but obviously the best way to remove it from the environment is to no longer add it to tires. So until that happens, we're going to be wondering how big of a problem is this in Alaska. So I hope to have some more results to share after this coming spring and summer. If you're interested and if you think there's any areas that are outside of Anchorage, obviously there's a lot more of Alaska than Anchorage. Anchorage does have higher road density, but we don't have any data from outside of Anchorage currently, so I would like to get some. The trick is just the timing is really important. I don't expect to find 6-ppdq in rivers and streams, even on the road system when it hasn't just rained, because yeah, there is some dust deposition. It's windy and the road dust blows into the stream, so you get some 6-ppdq through that, but you're not going to get a lot compared to when rain kind of washes everything off the road all at once. So if people collect samples for us, they really need to be timed well with the rain event. The one tricky part of this is one property of 6-ppdq that makes it kind of less available to salmon in the environment is it's really sticky. It will stick to soil and plants and things like that, but it will also stick to sampling equipment. So we have to be really particular about what we use to collect samples and how we filter the samples to remove particulates. So we've done a lot of work to kind of optimize that. So I have the sampling equipment, and we have some funding to share with others. But if anyone's interested and thinks they have a place that might be worth sampling, then I would love to hear from you. You can reach out after this. Otherwise, we are actively seeking funding, which I just had a meeting this morning about applying for some more funding for tackling and working towards a solution for this problem. So I will wrap up there and take any other questions.
Anybody have a question for Brian? Brian, I have a question for you.
Yep.
Are all of these studies, like you said, this was pretty recent that we found out this was a problem. Are all of these studies happening in Alaska, or did you say the first one was maybe Washington, if I'm remembering correctly? Has this been looked at anywhere else in the United States, maybe on like some more freshwater based fish?
Yep. Yeah. There's like every day there's new work coming out. That the original problem was discovered in the Seattle area, but certainly other places with salmon have been working hard on this. Like Vancouver was another kind of hotspot that had some early work. There's some work around the Great Lakes I've seen, and there's been some other random species, like a coastal cutthroat. I think was maybe one of them. Some other like a rainbow trout is somewhat susceptible. So there's been work all around the world that has looked into this, just to find out what else could be susceptible, or even in humans. Are humans affected at all by it? Obviously, we're not.
Sure.
It's not acutely toxic to us, clearly, because we're exposed to it all the time. But there's more and more work coming out every day. But I'm not aware of anything else in Alaska besides what we've been doing. So it's a big data gap.
I have another question. Has anyone done anything as far as trying to find a proactive solution? So like if it's photoreactive and it'll help break down, have you found out what frequencies are more effective in the breakdown, and then try concentrations of that frequency of light you can make some device and mount it on every storm drain in the state, and zap this stuff as it comes off the road.
Yeah. I mean, there's definitely been a lot of efforts that type of approach is common in wastewater treatment, and it would certainly be effective in that case. It is really hard from a stormwater perspective, because when the problem is worst is when the challenge is also the greatest, because the flow is really fast and the water is really turbid, really brown and full of particulates. And those two things together make that photochemical approach not very effective. So it shouldn't be a hard compound to remove, because like I said, it's really sticky. And like there's a lot of common, like your typical water filter, like a granulated charcoal type filter would probably do a fine job of removing it, but you can't really expect to flow all of the water that's coming off the roads through one of those at the rate at which it's running through the storm drains. And in that sense, it's a challenge. And all of the best solutions that have been found so far kind of are summarized in this figure on this slide of this green infrastructure kind of type of approach, where the water doesn't just run off straight from the road into the stream. It goes through some sort of bioretention cell, like some way that the water is slowed down and goes through vegetation and soil first, and that does a fine job of removing it. The problem is where you have roads that have the ability to concentrate all of the runoff in one small pipe directly into the stream, and that's what Anchorage has, and that's what a lot of cities have had. So it would be expensive to change that at this point. So we would love to have an engineered solution to just treat the water before it gets to the stream. That's still an area of active research, and I don't know of any good solutions yet, but we're definitely working on it.
Great, thank you.
Thanks for having me.